This procedure details the use of laser capture microdissection to isolate neuronal layers from the olfactory epithelium tissue and also outlines some downstream gene expression analyses. Start with a nasal biopsy to obtain the olfactory epithelium tissue. Then perform laser capture microdissection to isolate the neuronal layers. Next, isolate RNA from the samples, reverse transcribe for cDNA, and analyze for gene expression by qPCR. Ultimately, laser capture microdissection is used to show that an enriched population of neurons can be isolated from olfactory epithelium tissue and applied to numerous downstream applications.
The main advantage of this technique over existing methods like neuronal cell cultures is that it allows us to study both state and dynamic changes in a neuronal context.
We first had the idea for this project when we discovered the potential of combining laser capture microdissection with olfactory biopsies to obtain enriched neuronal populations. This method was established and demonstrated by Tejinda et al. We expanded on this methodology and combined downstream qRT PCR analysis in patients and controls. Demonstrating the procedure will be Yujin Chung, a technician from our team at Johns Hopkins University. The following procedures will be filmed at the Johns Hopkins facilities.
Immediately prior to microdissection, let frozen slides thaw for 10 seconds.